linkage in DNA. In the previous lecture, we have discussed about the structure of a DNA and RNA. What is the composition of a DNA and RNA? Till now, no exact structure, that which kind of the bases they are present, what is the bond which is present in the bases, that we do not know. Later on, with different experiment with the main which gave the double helical structure of DNA, that is a structure of DNA by Watson and Crick, that leads or they give a very defined structure of a DNA, right? So, first of all, let's discuss about the linkage in DNA. Overall, two type of linkage is present in DNA. What are those linkage? First linkage is called as a phosphodiester bond and second is called as a glycosidic bond. Let's first discuss about a glycosidic bond. So, what type of linkage? This glycosidic bond. And this glycosidic bond, you have to remember in this case, we have N-linked glycosidic bond. Glycosidic bond, and second is a phosphodiester linkage. Phosphodiester linkage. Phosphodiester linkage, right? First let's discuss about this glycosidic bond. Glycosidic bond is that bond, the name itself indicates glycosidic, that means a ribose sugar is definitely there. And this is a glycosidic bond which is present within the bases and within the ribose sugar. Ribose sugar and the bases, they are connected by a bond and the name of that bond is glycosidic bond. So, these are the glycosidic bond present in bases, present in between nitrogenous bases, present in between nitrogenous bases and sugar, right? And you have to remember which is there, N-linked glycosidic bond. In some cases the O-linked is also there, but that you no need to learn, but just learn, this is for your medical entrance examination point of view, that N-linked glycosidic bond is present in them, right? Now let's talk about a phosphodiester linkage. The name itself indicates the phosphodiester linkage. Phosphodiester linkage is that linkage which is present in the 5' to 3' direction. So, this is a present or so let's write about here, let's write phosphodiester linkage present in between where 5' OH of one nucleotide to 3' OH of second. Of one nucleotide to 3' OH of second. So, this is a phosphodiester linkage, right? Now, a very beautiful structure of this DNA is obtained. How this structure is formed? Let's see that. We know that the whole structure of a DNA is like this. Let's see how this structure look like. Which I am drawing right now is a ribose sugar. Ribose sugar is what? Which is having a 5 this carbon is present, right? In this case here the phosphate is present, right? And this phosphate is present at the 5 prime group. The 5 prime phosphate group is present like this, right? And in between these we can see that the CH2 is present. So for that I need to create a space. So C along with this H, and this is also another H is present like this, right? And here from this side, that is a another, that is base is present. So base is present like this, right? Similarly, these are joined. Again, this phosphate group is there, and this phosphate group is joined like this, like this. So this is called as a, this particular bond is called as a phosphodiester bond. Phosphodiester bond. Phosphodiester bond. So these are what? These are ribose sugar. These are sugar. Again, if you look at this, this is also a sugar. Again, they are joined like this. This is a phosphate group present, and this phosphate group is present like this, and they are present like this. So this is how a structure of a DNA it look like. When we look at this bond between a sugar and a base, and this base is present everywhere. Even in this case, the base is present over here. I don't have enough of space, so here also bases are present, and even in this case we have a base present over here. This is a depiction which shows that which type of bond, which or the linkage is present. This is a phosphodiester bond, and again which type of bond is present over here, which is called as a N-linked glycosidic bond. So which bond is this? N-linked glycosidic bond. N-linked glycosidic bond. So this is what? Structure of linkages in DNA. Right? Now, let's talk about, we have discussed about these two type of linkage. Hope these two linkages, they are clear to you. Now, when we talk about this nucleotide, nucleoside, what are these? Let's discuss about the nucleotide, what type of nucleotide and nucleoside is present in case of the DNA, what type of nucleoside is present in case of RNA. Similarly, when this nucleoside, they combine along with the phosphate, what will be the formation? So this is a quite basic thing, and you have to remember that as well. First of all, let's discuss about a type of nucleoside in DNA. So let's make a table for that. Type of nucleoside, and which nucleoside I am referring over here? That is in case of DNA, right? And what type of nucleotide will be formed, that I will discuss over here. Type of nucleotide. What are they made up of? Because students always get confused between the nucleotides and the nucleoside. Before going into detail, I would like to tell you about these things, because these are the words which I will be continuously using in the different the subsequent sessions. First of all, in case of the DNA we are talking about, that means the nucleosides will be there, right? Oh sorry, deoxynucleosides will be there. First of all, let's talk about, you must have heard of deoxyadenosine. Deoxyadenosine. That means deoxy is there, that means we are talking about DNA only. That means adenosine is that condition in which, what is there? We have one nitrogenous base present, and that is present along with a sugar. That means here we have one adenine, and that adenine is present along with a sugar. Right? Now, let's suppose something. If I join this with a phosphate, this is a phosphate. If I join with this a phosphate, what will be the formation? There is a formation of deoxyadenosine monophosphate. Deoxyadenosine monophosphate. So what we have learnt, that is deoxyadenosine, that is adenosine is there. That means adenine is present along with the sugar. When they combine with a phosphate group, what is the formation? That nucleotide is called as a deoxyadenosine monophosphate, and this deoxyadenosine monophosphate is called as a DAMP. So this is called as a DAMP, which overall we can call it as a nucleotide and a nucleoside, right? Let's take another example of deoxy. You can take an example of guanosine. Guanosine. That means guanine is present along with sugar. Guanine plus sugar is there. Now, if I combine the same with the phosphate group, now what is the formation? That is DGMP, guanosine monophosphate, and we can call it as DGMP. How this reaction happens, that we will discuss later on. So this is the second type of nucleotide. Now let's talk about the third type, and which is present in case of the DNA. You can take an example of deoxycytosine. Deoxycytosine means C plus S, right? C is present, this is a C, right? C cytosine plus sugar is there, and in this case the deoxy sugar is there. Now if they combine with the phosphate group, what is the formation? DCMP, deoxycytosine monophosphate, DCMP, right? Hope you can elaborate by yourself. Similarly, let's talk about the fourth one. We are left with a thymine, thymosine. Right? In this case, what will be the condition? The T is there along with S. When they combine equally with the phosphate group, there is a formation of D, T, M and P. Right? So these are the types of nucleotides which is present in case of DNA. Name the enzyme that transcribe hnRNA in case of eukaryote. Now, first of all, tell me the full form of this hnRNA. hnRNA is a heterogeneous nuclear RNA. And what is the full form of RNA? That is a ribonucleic acid, right? Heterogeneous nuclear RNA. Now, they are asking about the enzyme, and here the process which is mentioned, that is a transcription. How? Because they have written the transcribes. So that means they are referring to the transcription process. That means what they want is from a DNA, we have to form a RNA, and that RNA that is mentioned in the question, that is a hnRNA. So whenever these transcription happen, the enzyme is required which is called as a RNA polymerase. Because we are discussing about the eukaryotes, so the major enzyme which transcribed is called as a RNA polymerase 2. So this is a correct answer for question number 1, right? This is how you have to attempt. If you will write the RNA polymerase 2, you will get a complete 1 mark. Now let's move on to the next question, question number 2. So just read this question by the time that we prepare this board. So question says, how is repetitive or satellite DNA separated from a bulk genomic DNA for various genetic experiment? A bulk DNA is there and we have certain repetitive sequences present in case of the eukaryotic DNA. And majorly we have this repetitive DNA sequences present. These repetitive DNA sequences, they form, you can say that overall when we say the amount of these bulk DNA, they are very high. When we talk about the separation in the genetic experiment is done by density gradient centrifugation. Density gradient centrifugation. Density gradient centrifugation. So for question number 2, correct answer is a density gradient centrifugation. Let's move on to the next question, question number 3. So question number 3 is also a 1 mark question. If you'll be able to answer this, you'll get a complete 1 mark. So mention the role of codon AUG and UGA during protein synthesis. Now tell me one thing, another name of protein synthesis, that is called as a translation. That means they are referring to a translation process in which the RNA, they will form a protein. And this RNA is called as a messenger RNA. Now whenever a protein is formed from this, we need certain enzymes, we need ribosomes as well, and one of the important feature is that they require one RNA which is called as a tRNA. tRNA full form is called as a transfer RNA. Transfer RNA, they carrying certain anticodons, or you can say they carries amino acids. They carries amino acids. This tRNA, they goes to mRNA, later on they decode whatever the sequence which is mentioned in the mRNA, they are having an anticodon present in them and they are having an amino acid attached to it. And these amino acid, they are left over there, after that the uncharged RNA there is released. And again similarly the triplet codon is there which is being converted into an amino acid sequence. Now that triplet codon which is mentioned in the question, that is a AUG. AUG is called as a start codon. Start codon. This is that codon from where the translation always starts. Translation always starts with AUG. This you have to remember. And this is carried by a tRNA which is called as a charged tRNA because they carries one amino acid with it, and name of that amino acid is called as a methionine. Which amino acid? Methionine. Right? Now, this methionine later on they are attached, and further so many other amino acids they are added, and in between the enzyme is there which is called as a peptidyl transferase, which help in the formation of peptide bond between the different amino acids. Protein chain will be formed. Another codon which they have mentioned is a UGA. UGA. UGA is a stop codon. It is a stop codon, and this stop codon doesn't code for any amino acid. Doesn't code for any amino acid. That means whenever such type of codon they will come in mRNA, the whole translation process will stop there only. That the sequence of amino acid which is formed, which is attached to one another by peptide bond, they will also stop, and ultimately that amino acid chain, a long chain amino acid, that will be released, right. So AUG is a start codon, whereas a UGA, UAG, etc., UAA, etc., they are stop codon. So this is a role of these two type of codons. Normally there are 3 different codons which act as a stop codon, and we have discussed in detail in the sessions. Now let's move on to the 2 marks question. So state the difference between the structural genes and transcription units of the prokaryotes and the eukaryotes. Very good question, very important question as well. Yes, there is a difference in the structural genes of a prokaryotes and the eukaryotes. What is the difference between them? Difference is that when we talk about the prokaryotes, the structural genes are there. These structural genes, they are having a direct sequence that can be coded. First of all, they will transcribe them into a messenger RNA, and later on the translation happens and protein is formed. Whereas to talk about a eukaryotes, they are having a certain sequences which doesn't code for any protein. That should be removed. That is removed with the help of post-transcriptional modification, and that is called as a splicing. Hope you remember the 2 words which I am referring over here. They are called as a exon and introns. Exon are those which act as a coding sequences. In between the non-coding sequences, they are also present. These non-coding sequences, they are called as a introns, and they form a bulk. Basically, what is the function of these introns? They produces a heterozygosity, or they produces a diversity within the gene, so that a different combination of different proteins can be formed. Let's discuss about these. Now, the structural gene of prokaryotes. In case of prokaryotes, no non-coding sequences are present. No non-coding sequences are present. That means in them the introns, they are not present. So can I say no intron? Whereas when we talk about a structural gene of a eukaryotes, they are having a introns present in them. They are having introns present. They have a 2 region, basically intron and then exon, right. Introns are called as a non-coding sequences, whereas exon is called as a coding sequences. There is a difference in the significance of each of these. Even when we say the introns are there, and today we know they are non-coding sequences, but they do have a certain significance as well. Come to this side. Let's discuss about mRNA which is formed. Suppose this mRNA, they are having a certain sequences like this. They are having, suppose this region is there, then this region is present, after that this region is present, right. Now, suppose this region is called as a exon 1. This is called as a intron 1. This is called as a exon 2. This is a intron 2, and this is a exon 3, right. Whenever we talk about a translation process, before that all of these introns, they are removed. If I say introns are removed, then process is called as a splicing. A lariat formation occurs, spliceosome formation happens. So we will not go into detail. Now what will happen? All of these, they will be cleaved like this. That means we are left with only exon, exon, exon. This is a exon 1, this is a exon 2, this is a exon 3. Now the introns are removed. Now what I want, that these exons, they should be joined. When they are joined, let's talk about certain situations. Suppose they are joined like this, in which the sequence is 1, 2, 3, E1, E2 and E3. The type of protein which is formed, that is a P1. Let's talk about a situation in which there is a first E1 formation, after that the E3 is there and the E2 is there. When they will translate into a protein, a different type of protein will be formed, that is called as a P2. So hope you are getting what I am trying to say. A different kind of protein can be formed from a same mRNA. This is because of the presence of these intron sequences or the non-coding sequences which is present in them. So this is a significance of these introns, which is only present in case of the eukaryotes, which is not present in case of the prokaryotes, right? So hope you can write about this question, that is a question number 6. You can easily attempt such type of question. Now let's move on to the next question. Question number 5. Now, very good question, very easy, direct question. State the function of following in case of the prokaryotes. So, to talk about a tRNA, tRNA is also called as a transfer RNA. Transfer ribonucleic acid, and this is called as a ribosomal RNA. Ribosomal ribonucleic acid. Right? Now, tRNA is there, which is called as a transfer RNA. Transfer RNA, the function of transfer RNA is that they carry two things. One is called as an anticodon, and second is called as a amino acid. Right? Each of these, they are having a specific function. What are those functions? Anticodon, they are complementary to codons. And where these codons are present? Codons are present on mRNA. Right? So they will go and they will read whatever the sequence which is present in the mRNA, they will go and read over there. And because one specific tRNA is there and that tRNA, they carries amino acid, so they will go and they will give one amino acid. They will decode this and they will give another amino acid. Again and again they will give amino acid and an amino acid and an amino acid. They will keep on giving. Not the same type that depend upon the codon or anticodon which is present in them. Right? Now, and basic procedure, or if we talk about the whole procedure, they are helpful for a procedure which is called as a, we all know that is called as a translation. Translation, right? So what I want is now the amino acid sequences is there, out of these the whole protein sequences will be formed. My desired protein will be formed. Now just come back to this rRNA, that is a ribosomal RNA. So ribosomal RNA, they have two functions. One is called as they act as a catalytic unit, and second it acts as a structural unit. Hope you remember the translation process, where I have discussed the first of all the smaller subunit of the ribosome they come. When we talk about in case of the eukaryotes, let's take an example of a eukaryotes. In case of eukaryotes, the ribosome which is present is of 80S, and the prokaryotes, the ribosome is of 70S. These are the two different ribosomes they are present. So they act as a catalytic and the structural unit and which help in the process of translation, right. So we have discussed about the question number 5 regarding the functions of the tRNA and RNA. Now let's move to the next question, that is a question number 6. Where does peptide bond formation occur in a bacterial ribosome and how? That means they are talking about a peptide bond. How this peptide bond is formed? You know, when I was discussing about the protein synthesis, the ribosome is there. In the case of the ribosome, there are two sites. One is called as a P site and second is called as a site. P site is there and second one is called as a site. P site, whenever we talk about a first amino acid, let's take an example of a AUG or the methionine. When first amino acid comes, they always comes at P site, right? The second amino acids, they come at A site. And later on, whatever the uncharged amino acid is, that is released from a P site. And that charge, and they will left their amino acid with it. That amino acid will be bound to a tRNA which is present at the TA site. Now what will happen? This tRNA which was present at the A site, now they will shift again back to the P site. You know about the process of, I am discussing about the process of the protein synthesis. So they will again come back to a P site. Now the A site is empty for a new, you can say, the charged tRNA. Here I have discussed, once there is a shifting of these amino acid towards the A site, definitely they will attach themselves to the amino acid of another. What happens is that peptide bond is formed, and that peptide bond is formed between two, the COH group of first amino acid and again amino group of second amino acid. Right, peptide bond. And when we talk about the peptide bond, we can call it as a CONH bond. That is a CONH bond. When we write in a normal way, we call it as a CONH bond. Right, this is a CONH bond is there. Such type of bond is formed. This is called as a peptide bond. And the enzyme is also there, and that enzyme which is responsible for peptide bond formation, that is called as the peptidyl transferase. Peptidyl transferase. Right? This is how the peptide bond is formed. Enzyme we have talked about. We have talked about the two different sites. This is how you have to attempt this question. So this was a question number 6, which is also of a 2 marks. So even this question you have to attempt in a very short answer type. Don't write too much just for a 2 marks. So let's move on to another category. Here it is a 3 marks question. How many codons code for amino acids and how many are unable to do so? When we talk about a codon is there. We have discussed about these codes. In the genetic code, I have discussed genetic code. I have discussed that a triplet codon is there. Triplet codon is there. That means 3 nucleotides they code for a 1 amino acid. right? And today we know there are around 20 amino acid present. Though research is going on in this direction towards discovering the more and more amino acids as well, but we will be focusing on only the 20 amino acid which is already being discovered. Now the 20 amino acids which are present, they are coded by total 64 codons. And if I say a correct data, that is around 61 codons. Out of these, actually there are in total 64 codons. 64 codons are there. So basically, it is calculated with the help of a formula that 4⁻3. How? Because we know the chances that each and every nucleotide is 4. 4 different nucleotides they are present. This is a nucleotide. 4 different nucleotides that we know today, right? They form a DNA structure, and 3 because the 3 nucleotides are there which codes for 1 amino acid. So when we will calculate this, this will come out to be a 64. So overall the 64 codons are there, and these out of these 64 codon, only 61 they code for 20 amino acids. Whereas what about the rest 3? Rest 3 codons, they do not code for any amino acid. Rather than they code for stop codon. That means this is the place where whole translation stop. Translation stop. Right. What are these 3 codons? These are UAA, UGA and UAG. Right. They do not code for any amino acids. So these are called as a stop codon. So question says how many codons for amino acids are there? There are around 61 codons are there which is for amino acid, out of which the 3 are unable to do so. So this is the correct answer. Now let's move on to a second part. Codons are said to be a degenerate. Now, degenerate. This is one of the property of a genetic code, that is a degeneracy. Now, what do you mean by this degeneracy? Degeneracy means whatever codons are there, we have discussed that 61 codons are there, and these 61 codons they code for 20 different amino acid. They code for only 20 different amino acid. So if I say one amino acid can be coded by more than one codon, this is, we call it as a degeneracy of a genetic code. So one amino acid, because we have to code only 20 amino acid and we have a codon which is more than triple, not triple actually, which is a 3 times if I say, right? So one amino acid can be coded by a more than one codon. This is called as a degeneracy. Now another question says unambiguous. Unambiguous. Now what do you mean by unambiguous? That means one codon, they always code for one amino acid.